last class I have explained what are the evaluation of the drugs, and in that I have explained two methods. Now we are going to study about the microscopic evaluation. Microscopic evaluation means the evaluation of the drugs can be done through the microscope. That means the detailed examination of the drugs. Detailed examination of the drugs means which are the tissues present in that and what are the things present inside the drug, that we can study by the microscopic evaluation. See, as you already know, the microscope, it has the capacity to zoom the objects, and which cannot be seen through our naked eyes. It can be seen through the microscope. So here also the histological characters we cannot see through our naked eyes. So in that case, the microscopic evaluation of the drugs will be done by using the microscope. And here the thin sections of the drug are done, and the histological characters of the particular drug can be seen here. Histological characters is nothing but which are the tissues present in that and what are the things which will be present inside the leaf or the bark of the tree, that can be studied under the microscopic evaluation. That is nothing but the study of the histological characters. Here the first one, as I told you, the thin sections of the drug should be made, and the following points can be understood by using the microscopic evaluation. The first one is stomatal number. Stomatal number is average number of stomata present square millimeter of the epidermis. Means if you take a leaf surface and if you take per square millimeter of the surface, and here how many number of stomata will be present, that is considered as the stomatal number. The average number of stomata present per square millimeter of the epidermis. And this stomatal number varies from different species of the plant, and even the environmental conditions are also varying the stomatal number. And see, as I told you, stomatal number varies from leaf to leaf and environmental conditions. Example, if you take Datura stomanium, it is having the stomatal number of 87 in the upper epidermis, means upper surface of the leaf, and Datura inoxia, the stomatal number is 141, it is lower epidermis, that means below the leaf surface. So stomatal number, average number of stomata present per square millimeter of the epidermis, it varies with the species to species and even the environmental conditions. So next, after stomatal number, we go for the stomatal index. So stomatal index means the percentage of which the number of stoma form to the total number of epidermal cells. Means here the number in stomatal number, the number is considered, and here in stomatal index the percentage of the stomatal number is considered in the total number of epidermal cells. Means how many epidermal cells will be there, in that how many number of stomata will be present. That percentage of the stomata will be calculated in the stomatal index, and it is calculated by using the formula I is equal to S into 100 divided by E plus S. I is the stomatal index, E is the number of epidermal cells in the same area, and S is the number of stomata present per unit area. Here the example is Atropa belladonna. The average number of stomatal index is 6 to 10. 6 to 10. So stomatal index is the percentage of which the number of stoma are present in the total number of epidermal cells. So and each stoma will be counted as one cell here. Okay. So next, coming to the vein islet number. Vein islet number, it is the number of vein islets. See, if you consider leaf, here there will be the thin lines will be there. Those are nothing but the veins here, right? So it is the vein islet number is the number of vein islets present per square millimeter of the epidermis. Here it will be constant for the same species of the plant, and even the size of the leaf is not affecting the vein islet number. If the size of the leaf is if the size of the leaf is small or large, it is not affecting the vein islet number. So example is Digitalis purpurea. The vein islet number is 2 to 5.5, and Digitalis tapsi 8.5 to 16. So vein islet number is the number of vein islet present per square millimeter of the epidermis, and the size of the leaf is not affecting the vein islet number. So the next is the palisade ratio. Palisade ratio means average number of palisade cells beneath one epidermal cell using 4 continuous epidermal cells. This means, see, if you consider the epidermal cells here, so the 4 epidermal cells, if you consider these 4 epidermal cells and the cells which are present beneath is nothing but below, right? So the average number of palisade cells beneath 1 epidermal cells using 4 continuous epidermal cells. Means 4 epidermal cells will be considered here, and the cells which are present below that epidermal cells will be considered as the palisade ratio. So the example is Atropobelladonna, the palisade ratio is 6 to 10 and Datura stromanium is 4 to 7. So the palisade ratio is nothing but the average number of palisade cells beneath one epidermal cell using 4 continuous epidermal cells for the count. Means 4 epidermal cells should be considered as 1, and below whichever the cells will be present, that is considered as the palisade ratio. Example is Atropobelladonna 6 to 10 and Datura stromanium is 4 to 7. So hope you understood the microscopic evaluation. Next, another one is quantitative microscopy. We'll study it in the next video. Thank you.